Thank you, ladies and gentlemen. I would like to invite you all for this auspicious event, which is organized by Muslim trading agencies. One of our key principles, you can say that we are the sole distribution in Pakistan for binder climatic chambers. So, as a brief introduction, my name is Mohammad Kashif Chitani, and I am working as a business development manager for Muslim trading agencies. Let's start a formal opening of seminar by recitation of Holy Quran. I would like to invite my colleague, Mr. Abdul Rahman, our engineer, for the recitation of Holy Quran. Abdul Rahman, please.
أعوذ بالله من الشيطان الرجيم بسم الله الرحمن الرحيم لله ما في السماوات وما في الأرض وإن تبدوا ما في أنفسكم أو تخفوه يحاسبكم به الله فيغفر لمن يشاء ويعذب من يشاء والله على كل شيء قدير آمن الرسول بما أنزل إليه من ربه والمؤمنون كل آمن بالله وملائكته وكتبه ورسله لا نفرق بين أحد من رسله وقالوا سمعنا وأطعنا غفرانك ربنا وإليك المصير لا يكلف الله نفسا إلا وسعها لها ما كسبت وعليها ما اكتسبت ربنا لا تؤاخذنا إن نسينا أو أخطأنا ربنا ولا تحمل علينا إسرا كما حملته على الذين من قبلنا ربنا ولا تحملنا ما لا طاقة لنا به واعف عنا واغفر لنا وارحمنا أنت مولانا فانصرنا على القوم الكافرين صدق الله العظيم.
Jazakallah. Well, now we are moving towards the later session of this seminar. As a brief introduction about Muslim trading agencies, we are spending around 47 approximately years in the field of lab diagnostics, and we have like 30 years around for with the vendor, associated with the vendor. So I would like to call upon the stage the Vice President and Sales and Customs Logistics, Mr. Michael Puff, who came here from UK just to attend the seminar. So thank you, Michael, and please have a look.
Well, first and foremost, thank you MTA for inviting us to Pakistan. For us it is, for Adrian and me, who's my sales director for the New Middle East, including Pakistan, it's the first time to be here. And all I can say is an extremely great country, and we are hoping to come back on a frequent basis. I'd like to use the first 10 minutes to introduce Binder, because everybody maybe knows Binder, especially you guys know Binder, but you don't know who Binder is and what Binder represents. So therefore, this is the most important slide of the entire presentation that we have. It states attention to detail. Binder is a premium manufacturer of climatic chambers, among others. Attention to detail starts from us, not only from the engineering point of view and ends at the sales point of view. It has also the vacuum in between. That means for us, we take special attention to all the different steps that comes to selling a product and manufacturing a product. So, having that being said, being a premium manufacturer, you might understand as well why we are not the cheapest in the market. Binder as an organization is a SME. It's a 100% family-owned company in the first generation. And as a family-owned company, we focus on sustainable growth rather than fast and rapid growth with the consequences of jeopardizing quality or the customer satisfaction. So being a customer-centric organization and being a family-owned organization, we are very happy to also work with MTA, that is also a family-owned organization. Everybody has a boss, right? This is my boss. This is Mr. Binder, first generation of Binder GmbH, and he has given us clear guidelines to all the managers and all the employees. We focus on long-term growth. We focus on giving the best possible product, and of course, everything that we do is made in Germany. So you will never find a single Binder product that is manufactured or engineered outside of Germany. The Binder commitment. I will not read that and say nothing to it. But having been on the exhibition yesterday, having talked to some of the customers, it was very interesting to hear a common consensus among all of the customers. They said, selling is easy, and selling is rather easy. But keeping the customer happy is the most important part, because all the products in the world might have a failure. And it's now up to us, Binder and its partners, to make sure that the customers receive the best possible service afterwards. So our focus is on the best product, the best consulting, and the best service. And this is what you can expect from Binder, and this is what you can expect from Muslim trading as well. The mission. A mission, everybody has a mission. But for us, as an organization, the mission is rather simple. Giving you the best possible product. Why do we want to give you the best possible product? Because we want to make sure that we give you the best possible product for your application. Not the best possible option, the best possible product for your application. To make sure that we have a meaningful contribution to your work. A brief history about Binder. As I said before, Binder is not a very old company. It's now around 34 years in the market, and we have experienced over the last 34 years an annual growth of around 15%. What does this mean? We have started in an office that is not bigger than this here, right? And we set a clear target to be better than our competition. And better meaning better products, better technology, better sales, and better service. And this philosophy we have pushed through over the past 34 years. Today, this year, we have grown our turnover from zero in 1983 to almost 86 million euros, 400 employees, and 2 production sites in Germany. And we are intending to continue this success rate with satisfied customers as you are, and with great partners as MTA is. Before I was coming here, I made a little research in our documents. Right? And in 1986, Binder sold the very first container of products of B28, I assume, right, to Pakistan. So you see, we have not been growing in Germany first and foremost, but we have been growing in countries outside of Germany, right, including Pakistan. This is the reason we are so extremely thankful to be here, and to be so successful in Pakistan. Of course, for every organization, people are the most important asset. For us as well. We have 400 dedicated employees, and we Binder make sure that our employees receive not only the best training, but feel comfortable there as well. The average rate of average years of people at Binder are working for Binder is around 15 years, which is for a 34-year-old company quite high. Excuse me. Research and development. As an SME, of course, we are constant in competition with the likes of Thermo, Eppendorf, or any of the big multinational companies. The only way for us to be and to remain successful is research and development. We invest around 9% of our annual turnover in the research and development of new products. Not options, but products that help you to have a better result in your research or in your development. We have, of course, a vast expertise. You are all, or most of you guys, are manufacturers. We are manufacturers as well, and every single step of the manufacturing process we do ourselves. We are not outsourcing parts of the production or small parts of the production. Everything is done in-house, and it will remain in-house because it is a clear commitment to the quality and the products we give you to do your research. Lean production. All the pictures, of course, are from our manufacturing. We produce on an average basis 22,000 units per year. And all of these 22,000 units are made in Germany. And of course, made in Germany means a clear commitment to quality and to the best product. Excuse me. We have just finished the building of our new manufacturing hall. So this is a second one, which you see here on the picture, which added another approximately 5,000 square meters to our facility. Again, it underlines that we are clearly focused on our products, we are clearly focused on our customers, and we are clearly committed to the quality part of Binder. Binder is not only a climatic chamber manufacturer. We have a vast product portfolio. We have from ovens to incubators, to CO2 incubators, to high-performing ovens, to environmental simulation chambers, vacuum chambers, plant process chambers, everything in the portfolio. Please keep this in mind the next time you talk to your purchasing officer, right? Binder is present in 5 locations outside of Germany. We are present in these locations to make sure that our customers not only get the consultation from our partners, our distribution partners, but also from our own employees. We have around 100 employees in the 5 different locations: the USA, India, China, Hong Kong, and Moscow, to ensure that the customer, whenever they have complex requests or complex product requests, we can serve them. But also to make sure that our service expertise is transferred to the customer whenever needed. Because as said, it doesn't make sense to give consultation from the headquarters to markets such as Pakistan, but we have to make sure whenever our customers or our partners need us, that we are there for them. Our loyal customers, of course, we have many more customers, but the reason we have put these customers on the slide is very simple. Because these customers are strategic customers for us, where we have cooperations with. That means, whenever we develop new products, we give these products to select a few customers and ask them, can you alpha and beta test them for us? That means, is everything with that new product according to your specification, according to your needs? And the feedback from these customers we put back into the research and development and make them better. So this is, again, to underline how customer-centric this organization is and how centric it is to serve you, our loyal customers, in the future as well. So that was a brief introduction. Mr. Massoud told me yesterday that I have only 10 minutes, and you know, I am sales. I would like to talk more, even up to an hour, about my company. But now we are heading into the presentation of Mr. Adrian Stanton. Thank you very much for your attention.
Well, thank you, Mr. Michael, for this lovely and brief background for Benda. Well, now I would like to call upon stage Mr. Adrian Stanton. He is a product specialist and sales director as well for the vendor. He will talk about stability testing and ICH guidelines for pharmaceutical industries. Mr. Adrian, thank you.
So good morning. While they're setting up the presentation, I'd just like to also extend my gratitude to be here. It's my first time in Pakistan, and from what I've seen so far of this beautiful country, and the food is absolutely amazing. So I'm from the UK, so I experience a lot of this Asian cuisine, and yeah, it's one of my favourite foods. So just extend the gratitude. Absolutely fantastic to be here. So today, following on from Michael, talking about the company and the overall of the company, I'll be going a little bit more in depth now of the stability chambers that Binder produce, the real technology of the products, what it brings to you, what benefits it brings to you as a customer. And moreover, one thing we have, when Michael spoke about mission, he spoke about quality. We always have the same within our organization: quality over time. Okay? The first and foremost for any Binder product that comes to market is to make sure that that quality is of the highest standard, because we understand the actual market segments that our products go into. Okay? They go into industry, they go into hospitals, they go into university, they go into research work, and we fully understand that our product is critical when it's being used. Okay, so I think we're getting our presentation ready. Okay, can you all see the presentation now? Yeah. Okay, so I'll just briefly go over what I'll be discussing in a short time. This is a very long presentation, okay, so I've had to condense a lot of the information here. But what I'll try to do is give you as much information as possible, and then in a Q&A session that we have after this, if you have any questions, feel free to ask. Okay, so I'll give a quick introduction about what's being used in the stability testing. We'll look at some of the regulatory benefits and the basics, the chambers, the technical basics, and then we'll go a bit more deeper into the GLP, GMP, GCP standards. And then we'll talk about validation, because validation is very important, especially if you're exporting or you foresee to export your products outside of Pakistan. It's very important then you take into consideration the validation of the product. Okay. So, introduction into environmental simulation. Environmental simulation consists really of three elements: temperature, humidity, and light. And for us as a manufacturer, these three elements are very key to us. This is what we focus on when we build in our chambers, to make sure that we have the most accurate, reliable, stable temperature over long periods of time. The second is the humidity. Of course, we look at this, and I'll go deeply into how we produce our humidity, but humidity is very important as well, to have a stable, unfluctuating humidity over a long period of time as well. And a third element which we introduce is light. This is normally coming in the photostability testing side, because of course when these products are out in the market, they are exposed to sunlight, so I'll talk about that as well. So these are the three strong effects and the functionalities that we focus on within our product groups. With Binder we have letters that will represent the series of products. So just to give you a quick idea, because you will see these letters through the presentation, the K stands for the cooling side of the product, the B is the incubation/heating part of the product, and then the F is for the humidity. We also do an M, which is for material testing. Some of the functionalities that we have and features of the chamber when you look at this. So we have the red triangle, which is our trademark and what you see our identity in the marketplace. This is normally where the controller is positioned. And then you have on the left hand side as we're looking here, this is an option which you always should consider if you have products that is temperature sensitive. We have an over temperature and under temperature safety device, meaning if there's any spikes of temperatures, the unit will cut out, and if the temperature drops too low, the unit will also cut out there to make sure, or heat up, sorry, to make sure that your product is always safe inside our chambers. On the right hand side, these are just the buttons where you will switch on your humidity, where you can switch humidity on and off. You also have connections there for Ethernet and RS422. Okay, when we open the doors now, when you look inside the chambers, some very important key factors there. We have the ventilator at the top of the unit, but as you can see, we have three seals when we close the doors. Okay, we have a seal on the body, we have a seal on the inner glass door, and we have a seal on the outer door. Okay, so even in very hot countries like Pakistan, there is no external elements of temperature or humidities that can influence your product inside the chamber. It's very important to understand that. Also as well, we guarantee that there's no condensation running inside the chambers because we have a hermetically sealed chamber. And then as you look there, we have an access port that is standard on the side, which means that you can put your probes inside to do your own measurements. When we start looking at the build-up of the chamber, the inner chamber now, we go inside. Some key elements that I just like to go over. As you can see, we have the electrical heating around the fan, so we don't have any condensation build-up around there. We have the large evaporating cooling plates on the back of the unit. So this will give you a rapid temperature from heating down to cooling on a nice constant temperature flow. And then we have an evaporator plate at the base of the unit as well. Evaporator plate, what does that mean? This means that it collects any condensation that is inside the unit and it takes it away from the inner chamber. It's very important if you're using packaging inside there, you don't want this to become very wet, with condensation inside. So we have a large surface evaporator plate, and this will drain away any condensation water. And I'll go a bit more deeply into this later in the presentation. We have our new generation controllers now. These are touchscreen controllers. Okay, so this has been in development for many years now, to how we can bring a controller that is easy to use for a customer. It's self-explanatory, that you don't have to pick up manuals, that you can actually just navigate your way through the controller and learn very easy how to use that controller. Moreover, for stability testing, we also added some additional security features in there. Okay, so I'll touch on those as we go through this, but we have administration passwords, user passwords, and we restrict what functions can be used by each individual area. Okay, we have the RS422 interfaces on there, so if you're not using Ethernet connections, you can connect via RS422 and 485. And this is how we connect here on the Ethernet on the back of the unit. This is the performance of the KBFs. We have a KBF and a KMF. On the next slide I'll show you on the KMF. It has a wider and a broader range on the temperature and humidity factor. But this just gives you an idea of the quality that we produce our chambers in. They are very, very tight on the specifications regarding temperature variation. You can see there we're working on 0.1 to 0.2 and also on the humidity of fluctuation of 1.5%. Sorry, I think I went too fast. This is our KMF series. So here you can actually see now a larger, broader temperature range and humidity range. So this will actually give you a temperature range on the chamber from -10 to 100 degrees C and a relative humidity up to 98% humidity. Okay? And again, we have the very tight specifications that we work to there. Coming back to the home screen, so this is where I just wanted to touch on a few slides on the controller, because at the end of the day, the controller is the most important function that you'll be using. You'll be using your set point temperatures, you'll be using your recording facilities on there, and also your security features. What Binder do, which is different to other manufacturers that you see in the market, on the left-hand side on the picture there, you will see your set point temperatures, and on the right side, simultaneously, you will see the actual values inside the chamber. So you will actually see any fluctuation or any temperature drops or temperature rises simultaneously to your set point temperatures. Okay? The functions across the bottom there, I will go through individually now as we talk about them. But we have alarm functions on there, and also we have more information windows that go behind. The reason we started to put these controllers together like this is because in everyday's life now we're all using smartphones. And we're very comfortable with using smartphones, the functionality of a Samsung or an iPhone. And this is a very similar format now with our controllers. So once you've used them a few times, it becomes a routine process to easily navigate your way through our controllers. So these are the functions along the bottom, as we call them the icons. As you go through them, they all have a meaning. So you can see, you can change the screen, you can go back to your home. Like on your iPhones, you have a home button, it always takes you back to the main menu. And then you have your programs, your set points, your events list, what's gone on with the unit. You can clearly see what happened there. So we'll go into the first screen here. This is the main authorization levels. Very important for stability testing. Who has authorization to do what? Okay, it's very important. So we have 4 levels within this controller. You have a user level. Okay, they have no access to the settings or service within that unit. The second one is the administrative one. They have restricted access to the service menus. Then we have the service. So when MTA guys come in, they have a special access code to get in there, and they can see exactly what has gone on with that chamber. These chambers also do now like a self-diagnostic check. So it's very important when the engineer goes out there, he instantaneously knows exactly what's gone on with that unit. And then you have the master settings. Master settings gives you access to all areas of that controller. So this is what we tried to put together was when we're trying to think like the users now, and through beta testing and alpha testing, what pharmaceutical customers really want within their controllers, and they really want security and traceability. Okay, and this is what this controller gives you now. So as we go down to the next one here on the settings, in the settings menu, you have the chamber information. You can set the date and times. You have the display. You have your measurement charts. You have your variations, and then you have your interfaces. So there is a sub-menu that will run on the back of that, which is your Ethernet connections and web server. So now with these controllers, you can actually put in IP addresses, and if there's any alarms, if there's any issues with the unit, these now will send you an email. You'll be fully aware instantaneously you have an issue with the unit. Okay, so this is how it would look when you would go to your Ethernet connections. You put in your IP addresses, the company names, the administrator would sanction this, and then any problems that come from that unit, or if there's any data you want to take away from that unit, you can do this remotely. Okay, then we go into the program settings. Just to give you an idea of what we have there, we have 25 program settings within this controller, what you can do. We have 5 week programs. We have the programs that are done through the main menu settings, and I'll touch on those a little bit later. And you can literally start and stop them. Okay, so this is how it would look in your program selection modes. So very simple. We'll just go through here. You have your main screen on the left hand side showing your set point temperatures and actual values. And then you go through your sub-menus to start programming the units. On the bottom right hand corner here, you will actually see then it also has the chart recorder functionality. So you can actually see everything in a graphical format as well as a text format. For the service guys who comes in, these guys will come in and check all the data on a regular basis. Within stability testing, you should at least have downtime that you're able to actually service your unit and just run a routine maintenance check over the product. So this is where they would get the data from. They would actually see exactly what issues are with the unit, if it's been operating correctly, if it's been operating outside its parameters. All this now is recorded in the chamber. Contacts, if you ever have any issues, and outside of our distributor MTA, also you can gather the information from the controller, exactly where to contact us. Here, because it's touchscreen, we regularly, you can actually centralize it, so it's very easy to actually calibrate the touchscreen on there. So there's 4 menus on there which you can calibrate the controller. Just so for touchscreen, it's very easy to work. Any alarms, you'll have a nice red strip come across the top. It will give you the information exactly what that alarm is for. Okay, so you can see here it's highlighting temperature sign. So there's a temperature alarm that has been led away from here. Very easy to check what happened there through the main controller. The event list, this records everything that's gone on in the controller itself. So if you've changed the setting of your temperature or humidity, if you run a program, you have everything in a text format and it's date and time. Okay? This cannot be manipulated. So this basically is actual value, what happens live in the chamber. Here we're going to the chart recorder. So you can see here what we're running. So these are the parameters. You can actually see the temperature set points, and you will see exactly what's going on regarding on a chart recorders. These can also be exported from the unit into your documentation process. As you're probably aware, we increase the sizes of our climatic chambers. So now we offer a 1 cubic meter internal volume for high density throughput of product. This can also be seen today on the MTA exhibition stand. We have one of these products on show, so we invite you all to come down later and take a look at this. This is moved up from a 700 litre chamber. So this is a very popular unit now because it's the same size dimension as you look at the 720s, but it's much deeper now. This gives you a huge capacity for your pharmaceutical testing. Okay, photo stability testing Q1B. This is something that's very popular. We're seeing more and more now, not just in the pharmaceutical industry, we're seeing this in the food and beverage packaging industry, of now where they're introducing the light into the chamber. Within the Q1B standards of ICH, they recommend that you expose the product to 1.2 million lux hours of light, which is a mixture of visible light and UVA light. Here you will see on the controller it will display both, and also in the chart recorder you can see on the right hand side there you have the temperature, humidity, visible light and UVA light, so you can fully documentate and record exactly how much light your product is exposed to. Now that's not product with the LQC, we go one step further. We go from the light in the chamber as a whole right the way down to light recorded at product level. Because as you know, the further you get away from light, it loses its intensity. So we have the light quantum control, which is an option, which will actually measure the light at product level. And this can be documented, and then if FDA ever come in, you can say, we don't just measure the light in the chamber, we measure the light at product level. This is the light quantum control. So basically what you see here, we have the 3 light cassettes inside a 720 unit then. We use a horizontal lighting system, which gives the product full exposure right across the shelf. The light quantum control there, you can see in the bottom shelf we have these 2, best way to explain this is like T's with golf balls on top, and one is measuring the visible light and the UVA light. Underneath the light cassettes that we have a patented pattern on the base of the glass. And what this does, this disperses the light to 98% of the shelf area. Okay, so you can see the light source here is not just concentrated in the middle of the shelf, it's spread across the whole shelf, which means if you have product in the corner or in the middle, the light exposure is similar. On the right hand side you will see, let's say, a standard light cassette that doesn't have this patented pattern underneath, which means that you get the main light source in the middle of the shelf, meaning that has the most light exposure. With ours, you can see you get a 98% coverage of the shelf area. So these are the light quantum, LQC as we call them, sensors. These will be measuring the light underneath. So pharmaceutical industry, this is basically what information we've gathered over the years. As Michael showed you in the first presentation, we're very close with the pharmaceutical industry on a global level. We're working very closely with these guys on new changes, new applications, new research, drug to market. And we found that over the years, it takes average 8 to 12 years from product laboratory to finished product to the market. Development costs in a range of around $1 billion US, and not every product comes to market. You know, most big companies will be running 99 trials at once, and maybe only one of these ever come to market. So within that industry, we work very closely with 3 areas: the GLP, the GCP, and the GMP. Okay, so these are the good lab practice for GLP. Then we have the clinical practice, and then we have the manufacturing practice side of it. So on the stability testing side, I'll just be touching on 2 areas. What we look at in the manufacturing, which is the APIs and the FPP. So I'll be referencing those in my presentation as we go along. So API stands for active pharmaceutical ingredients, and then the finished pharmaceutical products, the FPP. So this is what we'll be touching on. And most of all for us, what we always feel, Binder will produce units, and we know that they have to be patient safe, because we know what's coming out of these chambers on a long-term stability trials. We want to give you the best possible product to make sure that you produce the best possible product for your customers. Okay, GLP, good lab practice. I don't think I need to go too much into this, but you know, as we look at the conditions, what we have the pre-clinical studies, and then we have the GCP, which the objective is the health side of this, to make sure that the safety and the well-being of the trials. And then we have the GMP, which is really looking at the quality assurance. The risk factors are very important. The validation and qualification, as I mentioned, of the equipment is very strict in Germany on our part as a manufacturer. We go through numerous stages of validation before our products come to market, and I think it's only right with pharmaceutical companies they use the same process then, that they're very strict with their own processes that they use. So we have 4 phases of the qualification I just want to touch on. So we have the DQ, which is a design qualification. We have the installation qualification, IQ, OQ for operation, and PQ for performance. This is a schematic that we actually put together here. I don't know if you can see the far left because I can't see it on my screen, but on the left hand side you would have the DQ and IQ, so you would have the master plan there. You look at your GXP, which is at GCP or GMP, and then you'll go through your URS, going down to your full specification requirements, your design specification, then your validation and identification installation. Those are linked on the right hand side then with your procedure testing, your documentation, staff training, your standard operation procedures, and then we look at the very end of the operational testing when you go for your OQ. So just to touch on these. So first of all, when you look at your API discovery, this is not regulated. This is the information that we get, that the API discovery doesn't have the regulation. When you go to preclinical development, this comes under GLP. Okay. Clinical phases 1 to 3 are GCP, and then when you go to market commercialization, again GMP. So this is the areas where we look at when we work with pharmaceutical companies about developing their drugs through our chambers. So the scope of stability testing, we already discussed that. Temperature, humidity and light are the key elements that needed to be used within a stability testing chamber. We're looking at the expiration date of the pharmaceutical product, which is your FPP, and then we look at the retest period of the pharmaceutical substances, the APIs. Okay, and both of these change under the influence of light temperature and humidity. ICH, I think we're all aware what ICH stands for, International Convention on Harmonization. Okay, these are the technical requirements, registration of pharmaceuticals for human use. Successful approach, I always underline this, successful approach for the industry and regulator in the EU, Japan, US. It's very important, quality, okay? That's what they're looking for, is making sure that you're using the right procedures and the quality of the unit. So we always establish guidelines on quality, safety, efficiency issues. The benefits of this is to actually reduce, or let's say reduction of your development costs, to avoid duplicate testing on a regular basis. If you don't have a product that works or it fails, you have to start all over again and go through the tests. So we understand how much resources pharmaceutical customers put into their stability test trials. We make sure that they have a unit that is reliable, that is there to work on long-term stability testing, okay, and that gives them everything that they require. They don't have to worry about the equipment, they can focus more on their development. These are the ICH publications on stability testing. A lot of us are fully aware, but these are the main ones, plus the appendix that go along with them. So we have the Q1A, R2, which is stability on new drug substances, then we have Q1B, which is your photo stability testing one. And then you have some or annexes, as we call them. You have the Q1C, which is stability trials for new dosage forms. And then you have the Q1D, E, and F has been withdrawn from the guidelines. This has been taken over by the WHO now. So these are just the overall appendix that goes through when we look at stability trials. The Q5C is stability testing for biological products. So Q1A refers to climatic zones. So these are climatic zones 1 and 2, but also there's more climatic zones which I'll show you in the next slide. So we distinguish 4 climatic zones to work in, and these determine temperature and humidities. So references and annexes of the quality guidelines Q1B, Q5C, we'll talk about those here now. So these are the ICH stability test zones. that we build our chambers around to make sure that we cover all of these global climatic regions. I would say for Pakistan it's normally zone 4B, which is the Asian testing conditions, hot, humid, which is normally 75% relative humidity, 30 degrees C. That just gives you an idea of the climatic test areas and zones. Formal stability studies. So again, the aim of stability testing is to achieve information about the expiration date, the retest period, the expiration date on the APIs, selection of the correct excipients, storage temperatures, packaging components, and handling of the manufacturer and transport of the goods. Long-term testing. So stability tests are recommended for storage conditions on long-term stability tests. However, these can be reduced by accelerated testing, and also if you have a problem with the testing on the accelerated, there's another one that's an intermediate testing, which we'll talk about a little bit further on. So stability test range for formal stability studies. On a 12 months, here we can see two different areas of 25 degrees C plus or minus 2 degrees and 60% relative humidity. And then when it's accelerated for 6 months, the temperature is risen to 40 degrees C, 75%. And then if you have failures or problems with it, you can do an intermediate test for 6 months, which is 30 degrees, 65% relative humidity. The other thing that's to mention here, as you can see, the first column here is about stability testing. The second column here, you will need refrigerators as well that can be used within these. And also freezers can also be used at a minus 20 test plus or minus 5 degrees C. So we're talking today about stability test chambers, but also we have other products that will support you along the way. So you have continuity of products that gives you reliable stability trials and stability testing across all areas. So here's a graph on the right hand side regarding our humidity range that we produce, and you can see from the studies here, long-term, accelerated, intermediate, all of those areas within the temperature and the humidity are in the white zone, which means that these are performed in a Bindi unit very nicely without any problems of condensation or not being able to reach temperatures and humidity there. Stress testing. Maybe some of you don't do stress testing, but we see this coming more and more now into stability trials. And this is areas now where they really put the product under extreme temperature and humidity as well. So here you can see this refers to the APIs. The test conditions are based on the accelerated stability testing trials. But you can move the temperatures up in 10 degree increments, normally starting around 40 degrees C. So here what they're doing is just literally seeing under very extreme temperatures and humidity how the drug, how the packaging really withstands itself within stress testing. So this refers to, with the photostability side, stress testing in ICH Q1B. The duration of the stress test is not specified by ICH. Usually we see in our discussions with pharmaceutical companies in Europe that it's twice the amount of the ICH dosage that they work to when they stress test. Typical tests on the thermal side are 70 degrees up to 3 weeks, and a climatic zone of between 60, 70, 75% relative humidity, also run over 3 week periods. And this is a degradation here of an API under thermal testing, thermal stress testing. You can see the degradation there over a period of 3 weeks when you stress test. On photostability testing, Q1B, again, I touched on this a little bit earlier. We work with the Q1B, specifies 2 types of photostability testing. Again, it forces the degradation of the tests. and it's to discover the pathway of the degradation. It's to clarify the safety and toxicology concerns of the API, and to define validate analytical methods for GCP and GMP. So also photosensitivity becomes a key factor also within the API, so we always look to see how the degradation and stuff like this goes there. So to conform studies, it's determine your storage conditions, obtain the suitable information for the packaging and the labeling side of the products, so the finished products, and determine the reconfirmation and the expiry dates of the finished product. We already touched on our LQCs here. This is what we're doing on option 1. You have the artificial daylight fluorescent tubes with ultraviolet light. We work very closely with option 2, which specifies 2 light sources in the Q1B, which is your cool fluorescent white light, and then also your near UV fluorescent lights. Okay, and this is what we conform to on the studies when we're applying option 1 and 2. Is the overall illumination is not less than 1.2 million lux hours of light. That's what they specify your product needs to have exposure to. The near ultraviolet light energy is around about 200 watts per hour per square meter. Okay, and the validated side of it, the chemical, ensures a specific light exposure. Okay, this is the activity during the drug development. We see the 6 steps. First and foremost, we'll just go back one, sorry. So first you have your API discovery, then you have your pre-formulation, your scaling up batch, then you have your registration batch, you have your follow-up, and then you have your ongoing. And all these fit within this area of clinical phase, preclinical trial development and commercialization to market. So again, just to go through the first part, API discovery, for our information, it's not regulated. Okay, it's only when you get to pre-formulation and scaling up, which is your preclinical development, you go to GLP stage. When you go to clinical phases, which is your registration batch, you're using GCP. And then when you come to commercialization, which is your follow-up and ongoing trials, you work to GMP standards. So these are the 6 phases as we talk about them. So stress testing with your API at a temperature of 25-70 degree, or as we said here, the hot and humid would be much higher, 30 degrees, 75 maybe, for up to 3 months. You differentiate the tests with your API, and then on step 3, you stress test 1 or more batches to determine the expiration date, the storage conditions, and the parameters for long-term testing. And then we go into the next step here on 4, which is your long-term accelerated testing, 3 production scale batches. Okay, that's both on the API and the finished product for a duration up to 12 months, long-term stability testing. And then we go into the commercialization stability test of 3 product batches again, and that can be a duration of up to 60 months, okay, on long-term stability testing trials. And then you have your annual stability tests with your API and your finished products. So just to touch on this, I know there's a lot of time is running out. Masood didn't give me too much time to discuss all this, but I just want to reiterate a few points. This is why temperature, humidity, dehumidification are so important within the chambers that you use. Binder go through rigorous tests in manufacturing. We actually go for a 27-point measurement check with inside our chambers. This is 9 points on the top shelf, 9 points in the middle, and 9 on the bottom. Okay? This is to our DIN standards that we work to, and factory standards. And we test between spatial and temporal to give you overall temperature fluctuation inside the chambers. And this is why we have such accurate chambers and reliable chambers. Within the chamber, when we start looking behind the exterior, we have the the air jacket system. so the APT line that we call it. And basically what we're doing, we create an air jacket around the inner chamber. So we have the heating elements. This is where the airflow comes around to have accurate temperature before it enters the inner chamber. And at every shelf position, as you will see on the product downstairs when you come and see this, that we have the air holes on each level of each of the shelves. And this gives a nice homogeneous temperature right across. So we have a very low air velocity, we have uniformity of the temperature, and I'll touch on the fan a little bit later because there's new areas that we conform to a medical directive there now. So dehumidification, very important. If you're moving from, let's say, a very high humidity of 70–80% humidity and you want to bring this down to 60%, you need to dehumidify this very quickly. Within our chambers you're able to do this, and the way we have that, we have the large evaporator plate on the bottom, and then in the base we have cold spots which collects all the condensation and water, and this is taken away from the inner chamber. So basically we create the dew point in the base of the chamber there. With our sensors, we use a very accurate capacitive humidity sensor measurement. This is a very reliable way of accurate humidity values inside the chamber. So the advantages of this, like I say, gives you a broad range. We can go up to measure up to 98% relative humidity, which is controlled. It's very precise, and also, like I say, it gives you very reliable readings. Within the steam module, okay, so we have purified water that comes inside our chambers. Okay, we don't use normal tap water now, we have purified water, and there's an advantage for that. So within our chamber we have a humidity steam module. This is constantly charged with a high temperature, ready. If you open the doors and you lose your humidity, it's ready to inject straight away to bring that humidity back to set point temperature very quickly. This is just a quick design of how the humidity module works. We have sterile steam coming in, which is reducing your contamination risks from the water, and of course very fast responding humidity values. Okay, we have two options, or let's say the standard chambers you can have plumbed to a normal tap water supply. Okay, and the water here in Pakistan is, I assume, is very hard water. So what we have a resin polisher that we put in between the water supply and the chamber. This is a resin polisher that polishes the water down and puts in purified water into the chamber. Alternatively, if you want to make your product portable, you don't want it fixed in one place, so you can move it around your production facility. On the right hand side here you can actually see two tanks. This is where you can actually pour purified water inside, and the tank on the left hand side will collect the wastewater. This can also be recirculated. Okay, so we can actually recirculate this. A lot of customers go for this now, especially on smaller chambers. If they have different areas of the production facility they want to do specific tests, it makes the unit very portable because we have lockable casters. You can move this unit around with its own water source. This is just a quick overview of the pure water aqua service that we offer within the chambers. As you can see on the right hand side, it has a traffic light system. So when the lights are green, it's showing that it's doing its job. It's purifying the water that's coming from your tap water supply. When it goes amber, this means that you should think about a replacement of the cartridge, or start thinking of ordering a new cartridge. When it's red, it means that your water quality is gone. It means that the quality of water going into the chamber is not good enough anymore. Okay, so it just gives you a very clear view of how your water quality is going into the chamber. Just a quick look of what's inside when you look at the polisher there. So as we go in, we have the water coming in. It goes through pre-filterization, then it goes through an ion exchange, resin filters, and then it holds all the particles there. So you can see it takes out purities from tap water and making sure you have nice clean water supply into the chambers. Something new for Binder that we made a change in our chambers, that we work to a machine directive now, which is a 0642EC, and basically this means that nobody can put their fingers now into a fan housing. If they're working inside the chamber, it makes it more safe when the fan is moving, that nobody can put their fingers inside and have the ends of the fingers damaged. So this is on the KMF range, and on the back of the unit, as you would look at it, we changed it from the left hand side to the right. Now you can see there's a solid plate there on the back. You can't actually see the fan, and there's no impurities can spurt from the back of the housing of the unit. So no hazardous things can come from the rotor at the back. So just talking about some advantages of Binder, these are two pictures here how we heat up with the advanced preheating technologies and how we cool the units. So in the center we have a couple of diagrams. You can see that the Binder humidity recovery is very, very quick. And on the base on there in the middle, you can see with our controllers, we don't have these fluctuations like this waiting to come to set point temperature. It goes straight up to the set point temperature and it holds a straight line to make sure that you have stable temperatures and you don't have any temperature overshoots or they don't undershoot. Okay, coming on to the DQ, IQ, OQ, and PQ. So this is now we're talking about design qualification describes the needs of the unit specifications. The installation qualification ensures the correct installation is done and that the operation of the unit is correct. And then we have the performance qualification, which compares the performance with the specifications. So here, following the steps of how these are done to GLP validation, we have the planning stage, which is prepared and written on a validation plan. Okay, this is assisted with the customer. Then we have the specification, and we agree on the requirements of what specifications you're working to. Okay, so this information comes from the customer. So we work very closely with you as production facilities to make sure that we work there. Then we have the test planning phase, which is the IQ, OQ, PQ. We have the testing phase, and then we have a review. And all this is done on site with our engineers here in Pakistan. They are fully trained. These guys will come in and support you every step of the way through your IQ, OQ, PQ on the Binder chambers. Okay, so the following steps are necessary as well when we start looking to GFP validation. So here, again, it's all assisted by our Binder partners. You can see we have the consulting side that we have. That can be done with Binder directly or through MTA. And we will work with you step by step. Very important for FDA. It might sound a bit stupid, but they really insist on this. That the documentation process has to be done in the correct way. So you will need a blue pen and a ruler, believe it or not. And the stamp is optional if you want to put your own stamps on there. However, what they say is every signature that is done by the engineer and confirmed by the customer has to be signed with a blue pen. If there is no data that needs to be put in any additional comments, a straight line has to be put through with a ruler. So this is how it would look in your documentation when you're doing this. And this is, FDA are very, very strict on this now when they come to do checks. So this is just something I wanted to bring to the meeting, just to understand when you are doing the validation to make sure that you use the correct way and methods, and then it doesn't cause you any problems later on. Okay, just a quick question. There is normally, can I verify the technical data in the OQ and PQ? You can do this, yes, in the OQ, but not in the performance qualification. The OQ is performed in an unloaded state, so an empty chamber, which is defined with the ambient conditions of your labs or your test area. The PQ is performed in a loaded state, so this means with all your product inside, under defined conditions, which is normally customer-defined conditions. Okay, so the process to start, the description is the detailed workflow, the description of the loading, size of the unit, the number of shelves, number of products, number of racks. Measurement points, how many measurement points are you doing? Are you doing 3, 6, 9, 12, 27 measurement points? The process parameters, which is normally your temperature and relative humidities you're working at, and then the time plots, the values against time, and the description of any other tests that you may be doing. So, in summary, because I know we have a Q&A session coming up now, Binder provides the KBF range that are compliant. The advantages of buying our product, they are internationally compliant products, especially for ICH testing. Binder KBF stability chambers, they're most user-friendly. I already spoke about how we've changed the controllers now to make them very user-friendly. You can actually use these without touching a manual or reading the manual how to program. Virtually there's no risk of deposits of corrosion inside, because we're now putting purified water inside the chambers. We pull out any impurities through the resin polisher. We conform to the medical directive, as I said now, with the fans inside. So again, less risk of any accidents there. More service friendly, lower maintenance costs long term. So again, the MTA team are fully trained. They offer an amazing service on our products. So if you ever buy a Binder chamber, you will have the guarantee of the manufacturer that MTA will be there on site to help you all the way through your steps. Fully compliant, so we offer additional validation, IQ, OQ, PQ, DQ. So this comes from Binder as a manufacturer. So this comes with the manufacturing of the unit. We produce the IQ, OQ and DQ documentation at the same time. And again, I just want to revert back to the final thing that we see within our organisation. We produce quality units, okay? Yes, there's time factors. Yes, the world today has a lot of pressure on bringing product to markets, units to markets, production facilities. However, we don't cut corners. We really make sure that our products, when they leave our production factory in Germany, are 100% reliable, 100% accurate, and they go through a string of stringent GMP testing before they leave our production facility. So that means when they arrive on the long journey here to Pakistan, they are in the same quality condition as when they leave our factory. Just to finish off, I hope you can see that there's a lot of customers, but we have a lot over the period of time, pharmaceutical reference customers here in Pakistan. We're very proud of this factor. This will continue, I'm sure, over the coming years, and we're very proud to be present across all of Pakistan, especially in a lot of these organizations that are not multinational, they're not global, but they're also local manufacturers, small local manufacturers, just moving out into the stability test world. And this gives us more pleasure than sometimes, when we see a new startup company starting producing with Binder chambers. So I'd just like to say thank you for your attention, and if you have any questions, I'll be available all day today on the exhibition stand. We can go through some of the features we discussed today in front of the chamber, and I think we're also with our experts here on a panel for the next 35-40 minutes. So thank you very much.
Thank you, Edgar. Well, before inviting my new guest speaker, I just would like to give him the brief introduction about himself. He is the former Director Health Canada and Drug Information Association. He spent more than 45 years in the regulation field regarding pharmaceutical and other. So you can say he's a living expert. And he's also the recipient of high recognition awards from USA. He's a member of ICH Expert Working Group, also the consultant for USB, WHO, UN, World Bank and others. So he will talk about challenges of conducting stability studies for biological and biotech products. So I would like to invite Dr. Sultan Ghani. So please come forward.
Thank you very much. Let me first thank the organizer of inviting me. And I think the most important thing when we look at about the history of stability testing in Pakistan, it reminds me, and sometimes it haunts me too, that it was the year of 2000. At that time, the association and industry association is quite a bit against performing stability studies of the generic product. So PPMA was totally against that. There is no need to do stability studies because the innovator has done it, so what's the use to do the stability for the generic product? And at that time, Dr. Farnaz was the Director General, and she asked me how we are doing it in Canada as a regulator, what we are asking, and I said that all the generic products, they have to do the stability study. It's a requirement. And that was the time the ICH was considering to harmonize the stability testing guidelines. So that was the year of that. Well, it was a little unfortunate that my suggestion or advice to Dr. Farnaz was to start gradually. Don't immediately ask to my data. Go part by part because industry has no chamber. Some smart scientists in one or two companies started saying if we can have a room, and that room can be controlled with temperature and humidity. And I'm sure most of you know which company I'm referring it, that really made a room instead of buying the chambers and do it, and claiming that they can control humidity and temperature within that room. So why to spend the money and buy the chambers and all that? So that is anyway very ridiculous idea. You know, you have to understand the whole science, and now you listen the beautiful lectures, all the details, the complications that involve in controlling the humidity and the temperature. Well, it took almost now, I should say, it took almost 17 years, 16, 17 years now, and we can see still that out of maybe 700 pharmaceutical companies that are existed here, maybe 100 companies are started doing some stability studies and realizing it's an important component. They are not doing it, none of the company, none, that's my statement, is very firm. None of the company is doing the complete stability testing as required by ICH guideline. That's my statement. Out of 700 companies in Pakistan, when we talk about the stability study and you are manufacturing 60, 70 products, you have to do the stability of 60, 70 products. That's not your choice that you pick 4 products or 5 products. And this is because in last year there was I was asked by Punjab government to enhance and improve the system of drug procurement. And at that point I said that there could be 2 ways that you can assess the company. One is to perform the inspection, or secondly, you can do theoretically. So the management was very much surprised and asked me how you can do theoretically. I said, well, you have 2 months, 3 months left. How you can go and inspect 200 companies? Well, they can, at least you can go and visit and go through it and maybe 2 hours, but that's not an inspection. So my suggestion was agreed by the tertiary group, and then they said that that's fine, do it. So I designed a protocol and I asked 3 key questions. And I said I can assess a company on these 3 questions about how good they are, how good their compliance is. Number 1, that do you perform complete testing of API? When you buy it, the API, do you perform it? Yes or no? Very simple. If yes, then give us the evidence. Do you do the vendor certification? Yes or no? And everybody said yes. Not a single company, no. And none of them can produce a specification. Out of 300 applications that I reviewed, 60, with all the difficulties, that at least there should be 60 companies that can supply the drug to the government of Punjab, was qualified. My last question was stability. Do you perform stability? Everybody said yes. Well, if you perform this stability, please give me the size of the chamber or the number of the chambers you have it to perform. Now the product list was already there for 300, and the size of the chamber was a small chamber here, less than the one in the picture. So with this one chamber you can do 300 products. This is all nonsense, you cannot. So I think that is quite an important. There is not much difference between to perform the stability study of a biological compared to the pharmaceutical. But there are some challenges to do the biological testing of a stability study when you do and compare it with the pharmaceutical. Pharmaceutical is much easier. And I can tell you that the ICH guideline of stability testing is one of the best guidelines. It was involved not only, and I don't call myself an expert, I was part of the guideline, but there were experts. There were real experts, those who have spent their life all in stability testing, and they are the part of that guideline. They wrote it. They understand the whole kinetics of these many molecules and understand it, how the stability works. So I would say that, and I insist that not to write any guideline. Don't make kind of a mockery that you have already created, that every week you are doing the stability. Nonsense. You disrespect the science. So you have to follow the guidelines that have been written by experts and you use it. And you can say that possibly we will do 5 products and we will do next year 10 and 15 and 20. That's fine. But do them properly. Not like every week you are doing a stability study. That's a joke. So I said it too. I'm not afraid of that, but I told RAP that don't do that. But I think still the companies are doing it. Those who are making some new molecules, the size of the product is about 500 capsules or 1,000 tablets. That's the size, which is again so ridiculous. Can you make a meaningful calculations between the development of 1,000 tablets and capsules and then make a bag of 500,000 tablets? No relation at all, no science that can support. Anyway, with that, I'll start. I won't talk too much with the time. I think I would prefer that we should have more questions. I do a lot of training, and I have done so much training that sometimes I feel that the traditional training should be abolished now. This is my own concept. And giving training by, by USP, World Health Organization, all this, giving lectures and all those, that type of the training is no more meaningful. What should be the way I think the training is that you put the subject and somebody should introduce the subject, that this is the subject, this is the total discussions of on this area or the research on this area, and let's talk and let's ask questions and let's raise that, and that type of the training will remain very, very good, just rather than reading those guidelines or reading those textbooks and saying that this is what is that. So I think it is rather very difficult that we should look into that. So I won't read it all my presentation. It is available. I'll give it back to that. Basically, I try to highlight few points of the biologicals that are a little bit difficult. Viscosity is an issue. The administration is an issue. The concentration is an issue. The way you deal it with that, the volume is small. All those are the challenges in comparing with the stability of a normal pharmaceutical dosage form. So that is the difficult. Then being the nature, they are the proteins, amino acid linked together, proteins and peptides, then they create another challenge. When they degrade, they become denatured. They won't degrade like a typical degradation of a pharmaceutical product. So you can easily determine the pathway of the degradation. Here you can say, while it's degraded, it's degraded. Denatured, it's denatured. So those are the key differences that you will find it. When we were having a little problem of the computer, I said, that's great, I can speak it with few points and the notes, and that will be the great, because I would rather prefer that we should have a discussion, we should have a question, you should bring your examples, and that's how it works. Because even in my experience, even many of us have reviewed and read the ICH guideline many times, and many of you are much more expert in performing these stability studies. You may find it that there are still the problem on the last guideline. There are two guidelines in the stability are. One is when we are talking about the bracketing and the matrix. That is a little more confusing because we think that bracket and matrix is so easy. I have got 5 different strengths. I can do it that way, or somebody else is doing some other way. I have a different size container. I can do it different way, put the high container and lower container. But we are not realizing that there are issues. Doesn't matter, irrespective of the fact that the container is the same, whether you are using class 1 or you are using class 2 class, but it is different. When we are moved in the plastic side, it creates more challenges to us. And that you will be surprised to see. Now, another problem that most of the North America and Europe possibly don't have it because they are not in zone 4B. For us, there is a problem. Because, and I will ask this question actually to you, what would have happened if your accelerated stability is showing more than 4 to 5% degradation. What will happen in your zone 4B when this is happened? Will you reject the stability testing? Will you accept that stability testing? What will we do? Or when we do, and the stability testing, when we do the freezing of these solutions, you know, that technology and that type of the practices are very common in North America, both in Canada, Europe, and in US. So what you will do it? So I saw somebody send me some stability to evaluate. I saw that the accelerated condition, the stability was showing between 4.5 to 5.5 degradation at accelerated at the point of 6 months. And the regular long-term stability, which is 30 degree at 65% relative humidity, is fine. It is not showing anything. And it was great till 6 months. And actually they got up to 9 months result. And it was okay. Now what you do and where you put the expiry date, how you put the expiry date. Because you have to do it. The expiry date is decided in your pre-market evaluation. Remember that. Otherwise, how you are going to market the product? So that's why people were asking me, usually when we are talking about regular stability study at 25, and then we will have accelerated, we have an intermediate of 30 degree. So if accelerated fails, we perform intermediate. Because all of you receive about 12 months extra, right? So our pre-market requirement is 12 months regular stability study, 6 months accelerated. If your stability is fine, you will get 24 months expiry date. That's the way it is. So I think we have a challenge now. We don't have an intermediate now. So those are the things that are quite important. Now the other thing is that is quite important is that Dr. Wad has recently started, this ICH training program. It's great that so I think that is quite an important that he is actually providing you that opportunity to know more about the ICH guidelines. I can tell you all of these guidelines are very difficult and people just think it, that no, we know it. Now you understand today in this presentation, let me try to close it because we decided to have more discussion and so that more interaction and you can bring your program. I think I'll just say a few words about these chambers. You hear, I think it's an excellent scientific presentation. It's not a marketing presentation. Whether you buy it or you don't buy it, that's not the issue. Okay? The point is, many of you or many of us, and at least since I moved to Pakistan and I worked with few companies, I find it major problem of these stability chambers. And sometimes we were wondering why. Whether we should trust that stability where the chambers are giving alarms and getting out of temperature or humidity, maybe in a month 5 times, minimum. Now what will happen? There's always a problem. And the company were hiding before the inspection. They are not reporting. They are making change, not doing the change control. Because they call these supplier, I should not repeat the name of the country. And the supplier is coming after 1 month and is saying that, okay, we should change this. I have never seen a change control written in a company. How you can do the quality management system without producing all these change controls that you have done? They have an impact. When you do the change control, and the change control has an impact, you have to do the revalidation. So because you don't want to do it, you are avoiding it. And you are saying, how many change control you have? The inspector comes and asks you the first question, how many change control you have? You are making about maybe 300-400 products, or maybe 100 products. And you said, we have 3 change control in a year. Impossible. We have 20 deviation. Impossible. Impossible. One of the inspector told it, I don't believe it. Because you have not written. The problem for us is that we all have to be right with all honesty and integrity. All the information we have it. We should not worry at all something wrong. Yeah. So today you hear what I learned it a lot on these presentations about the DQ. We never work it on DQs. When big people came for inspection, they are asking, have you done the DQ? The people are saying, you know, when we did the DQ, you haven't done your design qualification. Yeah, we did IQ. The guy came and made a footpath and put this. So anyway, with that, I'll close it. But remember that the presentation was very good, and when you put the quality equipment, you will have less problem. That's all it is. Thank you.
Thank you, sir, for this lovely talk. Well, now we are moving towards panel discussion. So I need more interaction from your side as well with our panelist, the expert. This panel was heading by our, as Michael said, that everybody has boss, so he is my boss, and my team leader, Mr. Masood Ahmed. Dr. Sultan Ali, you have a brief introduction about him as well. Dr. Ubaid Ali will join this panel. He is our PhD, member ISP in PDA, Deputy Director, Civil Services Office, Government of Pakistan. Thank you, sir. And again, Mr. Michael Hope, who came all the way from Germany, not UK, that's a mistake, from Germany. So we welcome him again. And Mr. Edwin Stanton, the Director Sales Builder, he came all the way from UK to just attend the seminar. So thank you and welcome. So please.
Assalamualaikum, my name is Masood Ahmed. मैं आप लोगों को उर्दू और इंग्लिश दोनों में चाहूँगा कि आप क्वेश्चन करें. सर यहाँ बैठे हुए हैं और आई विल ट्राई टू एक्सप्लेन एनीथिंग, क्योंकि हमारा मकसद सिर्फ इस सेमिनार का यही है कि लोग इंटरेक्टिव और सवाल जवाब पता करें. तो एवरीवन इज हियर. आई विल स्टार्ट दैट वी हैव सम माइक्स आल्सो. सो एनीवन हैव एनी क्वेश्चंस, सो प्लीज सारे लोग इंटरेक्टिव सेक्शन के लिए आई विल इनवाइट फॉर ऑल ऑफ देम. थैंक यू. एनी क्वेश्चन प्लीज. Yes, sir.
My name is Swalim Rizva from MTA International. Sir, I have a couple of questions. First one would be towards the vendor team. what is the standard life of the fluorescent lamp that you use in the chambers? The average life maybe.
Okay, I think I got your question. The average light. Shelf life.
The life of the lamp.
Oh, okay. Well, of course, we know we're using fluorescent lamps, so there is a deterioration over time. We work with the manufacturers of these lights. These lights will deteriorate, and they always say a check is normally done every annual year. But the answer to your question is around 2 to 3 years, depending on how continuously you use them. These can be easily changed, so there are replaceable light bulbs in there, so this can be very easily replaced. I would always suggest when you have photo stability testing is to really measure your light source, so you can really guarantee exactly how much exposure of light you have to your product. And this way, like I did in my presentation, the way to measure that, both visible light and UVA light, is through the light quantum control. It will give you exactly how much exposure your product is having.
You answered the second question as well, which was, how do you measure the intensity of the light when it's degrading with the passage of time? How do you regulate it? Do you maintain it, or is it like you just take the reading what's coming?
Of course, we can't maintain the light if it's deteriorating over a period of time. So we do the measurement on this. So when we're doing a long-term stability trials, when we're doing them in the factory, we will see the degradation of that light period over a period of time through the light quantum control. That can be fully documented within the controller, so you can actually see the degradation of that light over a period of time. And then it's down to you to make sure that you make the changes to those light sources to make sure that you keep a very stable light source on your product.
Great, thank you, sir.
Okay.
My next question, if I may, would be to Sultan Ghani Sahab or Dr. Ubaid. We heard about the portable units. That was the first time for me to hear about some. I'm sure there's a use for it. But would they need to be requalified every time they move to a new location?
In terms of installation, if you move it from that, once you have all done your PQ and things like that, if you are moving a chamber from some other location, definitely you have taken care of the installation side to make sure that it has been properly balanced, it has been taken care of those. Sure, you have to do that.
Thank you.
Ji, please, any question?
Hello. I'm Shahid from Prime Health Private Limited, Islamabad. I have two questions. First question is that do you have any software, PC-based software, by which we can monitor online monitoring of the data logging of the chamber?
Yes, we do. We have a GLP version of the outcome software, which is an additional software produced by Vinda. This gives you full opportunity to validate that software. It's 21 CR 11 compliant, so you can fully document everything on the chamber in a safe way with your software.
Okay, thank you. And another question is that there is any opportunity of sensors availability for the validation of the chamber? How would we validate the chamber? Is there any provision of sensors or at different sites within the chamber? Sensors, light sensors, or how many sensors are available for validation within the chamber?
Within the chamber, it comes standard. There's two. If you buy an LQC or as we call it, a KBFP, you will have 2 sensors in there as standard. However, if you want sensors for each shelf position, as a individual, which we have, we can manufacture that we have a light sensor on each shelf, which then is measurable for each light set.
I think Shaisa is asking about the temperature measurement or
Temperature, humidity, and light.
Light we have the quantum, which has already again shown you, but we can add it in every tray even. But for the temperature we have, and for the software even we are providing with, I think most of the companies have this software, and we will provide you with this next coming up instrument with the software. And we have a temperature mapping, and our engineers knows it's I think 27 points. Yeah. Temperature mapping
But just to add as well, I would always suggest, if you have the GLP version of the software, this should be validated as well. Okay, so please make sure that when you validate this software at the same time as validating the units, then you have a full documentation flow and full documentation process.
Cool. Thank you very much.
So my name is Mohammad Ali Rajput from Woodward. So I want to ask a question from Mr. Sultan Ghani. If batch size of our standard batch size is of 20,000 bottles, then how will calculate the batch size of a trial batch?
ट्रायल बैच
Yes.
20 साइज आपने बैच बनाया, ट्रायल बैच तो आपने पहले बनाया होगा। शुरू में CFR there is a rule, 10% rule was always there.
Yes.
What happened? It has been violated by the industry, because the industry is very smart way of getting confusing regulators. If they can just make a small batch of maybe 5,000, and they say the commercial batch will be 50,000, which is not. So industry decided, regulator decided later on to change. So WHO comes up and say, no, minimum 100,000. So now your batch size of the pilot batch is 100,000. This is the background behind it. Still the company used to do a lot of cheating. So that's the biggest cheating that has been done. A small budget, 5,000. Not the one that Trap is asking about 500. That's it. You just laugh on that. You make 500 capsules and then you make million capsules. There's a correlation somewhere? No, never.
Let's
clarify the word trial batches. We have two different terminologies globally. One is the pilot scale batch, and the second one is the lab scale batch. With the knowledge of the pilot scale batch and the lab scale batch, you move toward the commercial scale of batch. Right? The pilot scale batch is the 10% as a rule of thumb. Right? And two pilot scale batch, one lab scale batch, and the FDA just 2 or 3 years before decided what is the size of a lab scale batch. So we have lab scale batch guidelines and pilot scale batch standards, 10% of the commercial batch. We have to go on to pilot scale, one lab scale. And minimum is 100,000. If the pilot scale is more than 1,000, 10%, you will have to go upward, not downward.
So it means minimum 100,000 tablets are really you need for the batch.
For the one batch.
For the one batch.
You need 2 pilots.
Yeah, but for example, you are making peptides, and your batch size may be only about 40,000. That's your commercial batch vials. In that case, you can use the 10% rule.
If it is less than 40,000, it's
But I'm not giving word. This is just an example. You can use it. If your batch size is small, like peptides, a good example is peptides, or injectables. You are making only 40,000 injection as a commercial batch. Then you can use your pilot batch to 4,000, 10% of 40,000.
मैं एक बात और भी क्लियर कर दूँ इस मौके से, बताइएगा लोगों को, बताइएगा और उर्दू में इसलिए कह रहा हूँ ताकि हमारे भाई बस, अगर आपका टैबलेट सिप्रोफॉक्सिन आप बना रहे हैं सपोज और बैच साइज है 80,000, अब लैब जो हमारा पहले स्किल रिक्वायर्ड है वो 1 लाख का रिक्वायर्ड है मिनिमम, और 10% 80,000 का 10% निकालेंगे तो वो तो आप काट दोगे। तो भाई अगर 80,000 का भेजे तो प्लीज स्टॉप मैन्युफैक्चरिंग, डोंट गो फॉर अनलेस कि वो ड्रग ऑर्फन ड्रग ना हो या वो ड्रग रेयर डिजीज में ना दी जा रही हो, तो जरूरत नहीं ऐसे ड्रग बनाने की, बहुत से लोग बना रहे हैं। तो अपना बैच साइज जो साइंटिफिक स्टैंडर्ड्स है उसकी कैसे लिखे, लेकिन साइंटिफिक स्टैंडर्ड को मैथमेटिकली या इंजीनियरिंग के तरीके से नीचे ना लिखे।
My name is Yasir Yaqub and my question is कि जो biological products हैं, अगर हम उनकी stability testing कर रहे हैं और real time में the stability studies मतलब पास होती हैं, accelerated conditions के अंदर वो fail हो रही हैं, उसमें recommend ये किया जाता है कि आप intermediate studies use करें। In such case, should we market the product?
देखिए, अगर आपका ये है कि एक तो उनका जो एक्सेलरेटेड कंडीशंस हैं वो डिफरेंट होती है, जाहिर बात है, क्योंकि स्टोरेज कंडीशन जो रूटीन स्टोरेज कंडीशन है वो डिफरेंट होती है। रूम टेम्परेचर तो बहुत कम होती है। बायोटेक में अलबत्ता मैन्युफैक्चरर आर ट्राइंग टू मेक श्योर दैट देयर स्टोरेज कंडीशन इज रूम टेम्परेचर। रूम टेम्परेचर हम क्या डिफाइन करते हैं? रूम टेम्परेचर जो है बिटवीन 15 to 25 के रेंज में होता है, वरना तो आपके टेम्परेचर 8 to 15, 15 to 25 and then move up to 30 and then 40 and all that. So उस केस में देखिए रियल टाइम डाटा इज इम्पोर्टेंट। एक्सेलरेटेड कंडीशन ये तो दिस इज एन एडिशन, ओके, दिस इज जस्ट टू अंडरस्टैंड योर molecule. Otherwise, in most of the cases, the real data is the data. Okay? That's why they use the word intermediate. If a company does not want to do an intermediate abroad, this is the requirement. They don't have to do it. But they are not going to get 12 months extra time for the expiry date. So if you have got 12 months data, your accelerated condition failed, you do not want to do intermediate, what will be your expiry date? Your expiry date is only 12 months that you have the data on routine. That 12 months exemptions given to the ICH guideline is due to the fact that your accelerated stability is fine. There is no significant difference. And what is significant difference? It is defined as 5% or more. Then there is a significance in the degradation.
Let me add one more thing. There's two different words in the biological product. One is a refrigerated product, the other is a freeze product. For refrigerated product, ICH is included with the two different range of accelerated and real time. And for the freeze product, this is minus 20 degrees centigrade. Is for real time and for accelerated both. For minus 20 degrees centigrade of freeze product, we decide on case-to-case basis. And for the rest of the other biological products, which are the refrigerated product, we use 0 degrees centigrade or the 25 degrees centigrade as an accelerated case. In both cases, there is no intermediate studies. We believe on the real-time data.
And just to add to that point, normally the intermediate testing is normally done after the accelerated testing if you have issues. Okay, so that's where intermediate testing normally comes in, is after your accelerated testing, only with issues. Okay?
Yes, please, some more questions.
Yeah. I am from Woodward. My question is from Builder Expert. How long your Builder product can last, can run at preset condition in case of power failure? And another question that possible to design a stability chamber in one unit with different condition by providing the compartment in one unit?
Okay, so if I understand your question correctly, if there's a power failure, how do we maintain the conditions inside the chamber? I think this was your question. Yeah. Okay. Well, as I showed you in the presentation, we tried to make the chamber inside sealed as much as possible for outside influences. But of course, we have a fan housing inside the units. We also have the air inlets, which will, over a period of time, you will lose your temperature and humidity. How long that is, we would say that we could probably maintain, the temperature for probably, up to an hour or so. You will start to see this deteriorate because it's a fully stainless steel interior. We only use stainless steel inside out, which is a good conductivity of temperature, so it tends to hold the temperature longer and more accurate. But I would say an hour, you will start to see the loss of temperature and humidity.
Well, it depends on the ambient temperature, right? It depends where you have cooling inside, where the unit is standing. So 1 hour to 2 hours is feasible. Longer is not feasible.
And the second question is that can vendor has equipment in a different compartment have a different setting in one unit? Is it?
That's not possible. To compartment the inner chamber, I've not seen a chamber like this on the market before, where you can have individual compartment that is holding different temperatures, different humidity values. For us, this wouldn't make sense. I'd rather say go with smaller chambers and run a number of small chambers rather than going for a big volume chamber. And then you're able to run your different simulation tests in the smaller units.
Yeah, absolutely.
Sir, my और क्वेश्चन डॉक्टर साहब से और सर सुल्तान से कि क्या हम फार्मा फिनिश प्रोडक्ट जो मैन्युफैक्चरर है उनको एपीआई की स्टेबिलिटी टेस्टिंग उनको ज़रूरी है, कोई फार्मा गाइडलाइन या कोई गाइडलाइन है? और एक क्वेश्चन ये कि ऑनगोइंग स्टेबिलिटी टेस्टिंग के लिए नंबर ऑफ बैचेस की क्या कोई कंडीशन है? नंबर ऑफ बैचेस कितने स्टेबिलिटी पे रखने? लाइक अगर फॉर इन अ ईयर अगर 100 बैचेस बन रहे हैं तो उसमें से कितने बैचेस में ये स्टेबिलिटी टेस्टिंग करा?
देखिए, the question number 1 is your API stability. You don't have to do it because most of you are doses from manufacturer. If you are an API manufacturer, you have to do it of your own product. So since you are buying API either from China, India or Europe, wherever you are buying it, you should ask it the stability data. That is why we call it the open part of the DMF. That includes all the information of the API, including the stability studies. So that information is with you. You don't have to repeat, you don't have to perform. Okay, it's no legal requirement, regulatory. Anywhere, United States, Europe, Canada, anywhere. With respect to your post-marketing, in a way, after you have received the registration or marketing authorization, the regulators were very flexible. It all depends because they cannot write it in detail the exact number of batches you should put in. If you are manufacturing 100, you put 5. If you are manufacturing 5 batches, you should put 1. So they are very flexible. But in general guidance, at least 1 batch a year you should put it for your post-stability program.
Thank you very much. The scenario is if company is manufacturing 1 batch in 1 year, the intent for keeping any batch into the chamber or for stability is what is the intent behind? we want to have scientific support, and we want to have reasonable work and reasonable science. So you have to decide how many batches is required to keep on stability studies. If you are manufacturing 100 of batches and lot number of changes or lot number of divisions you have done, you need to put your batches on the stability chamber. The rule of thumb is one batch every year, but you have a valid reason not to keep on year because your product is stable since last 1 decade. You may extend the life, or you may extend your postponed stability program of particular batch.
Some more questions.
Question from the binder expert. What is the acceptance criteria of the water used in the stability chamber? You showed one of your slides. It's a deionizers is required for that. So what is the minimum criteria, or what is the conductivity of water is required for this?
Okay, because we're using around about 1 to up to 20 microsiemens of quality of water coming through to the chamber. The reason is we want to take the impurities away from the tap water that comes into the units. Hence the reason why we use these resin polishers. It gives a very good constant flow of purified water in there. Moreover, the reason we use a resin polisher is because it captures the impurities of the water coming through. So this means that the humidity value that you get inside the chamber doesn't really have any impurities or contaminants in there. It's literally pulling these. So we would say on the water quality, to answer your question, is anything between 1 to 20 microsiemens you will have of quality of water.
So it means that if any pharmaceutical is having this water level conductivity 1 to 20, they can use that water. Deionized water. Deionized water.
Deionized water. 20 microsiemens.
Yeah, after 20.
My name is Eva and I am from Pharmaceutical. My question is regarding biological products. What is the criteria for conducting force degradation on biological products?
Sir.
You know, before going to force degradation, we must know what is the stress studies. And we perform stress studies on raw materials API to know the degradation pathway of the product. And one is major intent to know the degradation pathway and the emission profile of the particular product or process. And the second intent to develop a method which can indicate any issue with your product or your materials. And if you have, for pharmaceutical, let me first compile the pharmaceutical. If you have stress studies in the API, there is no need to repeat in the finished product unless it is proving that the light may influence on the product. Is a rule of thumb, is a rule of understanding of the science around the ICH countries. The second is a biological product, the forceful degradation studies. And the biological product is a lot of variability. What is there? You must know your product may go wrong, so some indicator you may identify. That's why we identify different variables, and we try to identify different variables by the forceful degradation of the finished product. In finished product, we use word forceful degradation. Sometimes the literature use word stress studies for the finished product. But there is a difference between the forceful degradation and stress studies. Stress studies is usually used for the substances, and the forceful degradation is used for the product. And the biological, we need to demonstrate that our product is not degrading, and the pathway of the degrading product is not here. And sometimes in pharmaceutical as well. In the case of the 5%, there's a two different molecules of 5% and the isoniazid. We need to demonstrate this just by the forceful degradation studies because isoniazid and the 5% have an interaction. The 5% and the ethambutol have an interaction. One is a hygroscopic and one is the other way around. Both in a hygroscopic condition produces some degradation product, the other one in dry condition produces degradation product. But in some pharmaceutical, we need to demonstrate forceful degradation studies on the finished product.
Ji.
Assalamualaikum. I am Shumaida Khan. My question is regarding the long-term condition for Pakistan, as it is lies in 4, 8, 30, 65, but climate changes to hot and very humid. So the long-term condition would remain 30 degree and 65 RH or 75% RH.
From the guidelines perspective, and all these zone has been very much reviewed. It was just not written because the temperature are like that. All the considerations were given in the development of the guideline. You should understand it. The guidelines are not written in the ICH, especially the ICH guideline. There is a lot of research and development involved. So if they are giving you a guideline that it should not store above 30 degrees at 65 relative humidity. But excursion could have happened because in many of those countries we have a very poor transportation, we have a very poor distribution, we have a very poor storage condition, and we have sometimes at the retail very, very poor. So those excursions are there to a reasonable point, means half an hour, hour and a half or two, and has been taken care. Those excursions are being studied and are over. But for a longer period of time, those exposures, those excursions are not accepted. Your product will definitely deteriorate. That is the situation. You cannot just try to work it and say, well, we'll put on accelerated condition. So fine, certain molecules can stay. Potassium iodide, as long as you can do it within the acceptable storage condition, the product will stay 20 years. It will never degrade. So there are molecules that has that type of the stability profile.
So, sir, our question may be that either we can increase the humidity from 65 to 75%, is it? Or we will recommend only 65%?
You have to keep the same what has been written.
Okay. So I think you got the answer that you have to follow the guidelines, but the guideline has said you have to keep at 65, so you have to keep at 65%.
Just to add something to this, maybe you also see in the presentation that some customers now exposed to stress testing. This could be something where you could maybe, aside from just to see degradation that I showed you in my presentation, maybe that you can do a separate test, with very higher temperatures or higher humidity values for a period of 3 weeks to see if there's any severe degradation of the product. But again, if you're working to the stability guidelines, you have to work to what is preset by the ICH.
हम साइंस जरूर अपने बॉडी में लगाएंगे यहाँ पर, लेकिन सुपर साइंस पे ना जाए। वो साइंस पे ना जाएंगे, वो साइंस ना कि अभी देखिए अगर 30 डिग्री सेंटीग्रेड और 65 कहा गया और जोन बी के लिए हम 30 एंड 75, तो आप समझते हैं कि हमारा जो लाड़काना है वहाँ 50 डिग्री सेंटीग्रेड टेम्परेचर, तो हम 50 पे रियल टाइम पे करना शुरू कर। साइंस जो है ना भरपूर इसके पीछे एक नॉलेज बैंक, तो मैं आप लोगों को दावत दूंगा तो उस नॉलेज बैंक को पढ़िए कि हम कैसे पहुँचे 30 और 65 के ऊपर। इट इज सफिशिएंट। इफ़ यू हैव एनी इंडिविजुअल स्पेसिफिक केस जहाँ पे ये मामला हो रहा है, तो फिर उस केस को हम सुनने के लिए तैयार है, समझने के लिए तैयार है, उसके क्या है। लेकिन 30 एंड 65 इसके लिए तय कर दिया गया, उसके पीछे भरपूर साइंटिफिक वर्क हो चुका है। तो साइंटिफिक वर्क को पढ़ें, वहाँ पे कोई गलती है, कोई कमी है, उसको जरूर क्वेश्चन कर सकते
हैं। डॉक्टर साहब मेरा क्वेश्चन है सर, ये ब्रैकेटिंग और मैट्रिक्सिंग का जो कंसेप्ट है वो क्या सिंगल बैच के ऊपर एप्लीकेबल होगा? दूसरा क्वेश्चन ये, स्कर्जन का टाइम ड्यूरेशन कितना होगा? यानी कि अगर फेलियर हो जाता है किसी इंस्ट्रूमेंट का यानी कि चैम्बर का, तो स्कर्जन टाइम कितना है? क्या वो रिपोर्ट होगा हमारे उस प्रोडक्ट की टेस्टिंग के ऊपर, या हम चेंज कर लें उस प्रोडक्ट?
देखिए ये गाइडलाइन सेज इट अप्लाइज, सिंगल बैच भी अप्लाई करता है, ऐसा नहीं है, लेकिन उसकी कंडीशंस जो हैं यू हैव टू मीट द कंडीशन ऑफ ब्रैकेटिंग एंड मैट्रिक्स आर गिवन। ब्लैंकेट नहीं दिया आपको कि आप कर लें, उसकी कंडीशन है। मिसाल के तौर पर, फॉर एग्जाम्पल, इफ यू हैव ए फॉर्मुलेशन of 2 different strengths and the formulation is different, okay, it's not your formulation is not uniform, or then in that situation you cannot do the matrix bracketing. Formulation different हो गई. आपने अगर 5 strength है आपके 5 milligram, 10 milligram and proportionality है formulation की, you can do it. But if proportionality is not there, you cannot do it. The same is bioavailability. That's why we do the proportional formulation. People develop formulation differently. All there may be some reason, but you should try to have a proportional formulation so that you can do it 1 bio study rather than 10 bio studies. Okay, that's a fact. Same is in the matrix. So there are other conditions. I don't remember offhand, but there are other conditions that you have to meet it for the bracketing and matrix. So that my question was, this is what I want to ask here, that if for example the chamber, some reason, not the electricity, but some other reason they stop, there's some malfunctioning of the equipment, and that could have happened. If it has happened to say 4 or 5 hours, now you have to take out the samples, you have to work it out. In case if you have to rearrange everything, do you have to do the requalification, revalidation of the PQ?
Yes, the answer to that is yes. If you do have the failure or anything, you have to redo the PQ performance qualification.
Last question we want to take, so we have a lunch.
The one portion of this question was left unattended. The escalation you was talking about, during the manufacturing of the raw material to the finished pharmaceutical product, you hold sometime your granules, somewhere your granules, somewhere your tablet, somewhere cold. The cumulative holding time must not increase up to 5. If you increase up to 5.1, the stability studies will be altered. So that's why cumulative hold time should be less than 3 months. And if you was talking about the finished product, I will recommend you to go for mean kinetic time to find the USP to see all the skeleton impact on your diabetes.
Assalamualaikum. My name is Shabnam and I am from Pharmatech. My question is related to the force degradation studies, and I would like to know how much degradation is enough for the drug substances. About the 5%, 10%, 15%, how much the degradation we allow to?
The forceful degradation in API we consider as stress testing. In stress testing, once you started to procedure and after 3 months or 1 month you find it 30%, there is no need to go further. We are not developing a byproduct or the degraded product. 30% is enough. This is not written in any guidance on the rules, but as a matter of practice and as a matter of the experts who claim in different papers or different lectures, it is 30%. You have to stop it once you are at 30% degradation because you want to know the degradation pathway.
Yeah, I think if you remember in my presentation, you can see a degradation over a 3-week period. So normally after 3 weeks you should see the percentage, and like the doctor said on the end there, I think once you get 10, 15, 20%, then you just stop this then.
Assalamualaikum, I'm Sabah from Pharmatech. My question is that if we are importing our product to UK, so we'll have to do stability studies according to their phase, or our phase stability studies data is enough for that?
Stability study is the requirement of the government, and if you are exporting drug to UK, you have to comply with the requirement of the UK. And if you are importing drug in Pakistan, you have to comply with the requirement of the Pakistan.
It means still they have to do the stability if they are importing all the goods.
My name is Furqan. I'm from Pharmatech. My question is to builder people. Do you supply smaller accessories related to chambers like if we have issues in plungers or seals in humidity module of chamber, do we have to purchase the whole humidity module or only the plungers or sealers?
It depends on which series of the KVF you have. If you have the latest KVF model, we used to only supply the full humidity model, but as of September, you are able to purchase single parts of the humidity model, so you don't have to purchase the old one. I'm not sure when it will hit Pakistan because we have to train MTA for that, right? That they consult you correctly, but it should be doable within the next few months.
Just to add to that, the door seals, things like this, you know, we have the 3 seals on the outside. These can be produced and put on separately, so individually they can be changed.
Hello, Assalamualaikum. My name is Rehan and I am from 13th Pharma. मेरा सवाल ये है कि स्टेबिलिटी टेस्टिंग की जो विंडो होती है, कितने टाइम में उसको हम वो टेस्ट कर सकते हैं? जो स्टेबिलिटी स्टडीज जो हम चैम्बर से निकालते हैं उसके बाद कितने टाइम में उसको टेस्ट कर सकते हैं? कितना विंडो होता है हमारे पास?
Actually, there is no written guidelines for that, and you have to reasonably bring it to within the time limit that you are working in. You are not going to take the sample out and wait it and do it in the evening later on. I don't think that is. And you don't expect, that's one thing very much you remember, that I've been a regulator for 40 years. We don't want to give you all these things to tie you up. The guidelines are written. What? And how is yours? How you do it? So that is quite important. Many individuals want to know, okay, how many days then we will do it? Well, the regulator has given you quite a bit of flexibility to do it within a good scientific understanding. How should I leave it? For 5 hours, 3 hours, 2 hours? Like that. In a reasonable time, you take it out and you do it. You are ready to take the sample out. Possibly you are bringing it the temperature down. You know, the technology has changed quite a bit. When we used to do the IV solutions and we add the drug into it, we send it to the ward for the administration to the patient. Today, we make it before and put it in the freezer, and when the need comes from a patient, we throw it in the microwave and thaw it and give it to bring it at the temperature and then administer it. So the technology is changing, things are changing, so there is no fixed 2 hours, 3 hours after taking it. I don't think so.
We don't have any requirement, but we expect efficiency and we expect as you have some science behind it.
Hello?
Mr. Ghani, I have a question is there any criteria for surgical items, just medicated surgical items for stability studies? Same criteria for those? Medicated surgical items, bandages, etc. This type of items.
You know, once you apply any chemical agent which may go wrong under the conditions, we have requirement of stability studies. Because you're writing use until February 2019, we have to prove it.
Hello, Sanika. Hello, Sanika. My name is Umar Abdul Bari. I am from 13th Pharmaceuticals. I have a question to be asked for Mr. Ubaid Ali. Is it necessary that we have to perform our stability studies on refrigerator, on cool place? One, according to ICH guidelines. Second one, if our stability chambers becomes off, so we have to perform our stability right from the beginning or not?
The first question, if something is written in ICH without putting without explaining your question, if something is written in the ICH guideline, it is the requirement of the government of Pakistan. The second, you are asking about
a chamber, if sometimes it fails or off. So we have to perform a assembly right from the start or
no? No. You have to write the reason, you have to write the rationale, what you are going to do. If your rationale asks you to go for new product or for repeat, you have to explain it. It is not we who decide what you have to do. It is you, your coordination system, your thinking process, conclude on the basis of the science, on the basis of the guidance given in the ICA, given the different papers for your activities.
Everyone wants to add something.
Just to add to that, I think I'm right in saying within the ICH guidelines there is also downtime available for routine maintenance and things like this. The thing is, if you have a power failure, it's down for you. The responsibility is on your side to make sure that you documentate everything. Okay? This is a key factor that what they're looking for is why was the unit switched off or why was the unit down. And it's very important that you documentate and have a full traceability process for any power failures.
Last question.
My question is, can we consider the bulk testing as a zero-month testing of finished product? Can we consider this the bulk testing as a zero-month testing of finished product?
Not at all. Definitely not.
Because we have to put the finished product packaging material. Packaging material is the important critical material for this process.
Always remember, packaging component is the most critical. It is a decision point where you start. So your primary packaging is the, you cannot have the bulk and do the testing. Sure, your bulk, you are checking your bulk before you are packaging it, that's fine, that's okay. But you cannot do that. That you run on your initial assay on bulk and then you package it and the 3 months time is coming on the package material. So that's why the primary packaging is the key component and that will determine your expiry date, your shelf life, your things, all that. So that's very important.
Ladies and gentlemen, thank you very much for your questions. Definitely questions तो खत्म नहीं हो सकते हैं, but we have lunch time also. और हम available हैं hall number 3 में, the builder people and myself. And thank you very much, Dr. for your time and Ubaid. I'm really thankful for you people that you give us the time and thank you very much for the audience. हमारा lunch तैयार है. I think we should follow.
Please proceed for the lunch.
Thank you very much.